Welcome, everybody, to Wednesday Night Lab. I'm Tom Zinnan. I work here at the UW-Madison Biotechnology Center. I also work for the Division of Extension Wisconsin 4-H. And on behalf of those folks and our other co-organizers, PBS Wisconsin, the Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night Lab. We do this every Wednesday night by Zoom 50 times a year. Tonight, it's my pleasure to introduce to you Ross Meyers. He's the Director of Cell Manufacturing for the Program on Advanced Cell Therapy. Did I get that correct, Ross?
Close enough.
What does PAC stand for?
Program for Advanced Cell Therapies.
Good. And I'm going to ask Ross the 5 questions I ask everybody. Ross, you can answer these in any way that you would like. Where were you born?
On a farm south of Belvedere, Illinois.
Wow.
Not far from Dixon, Illinois, really.
Yeah, my heart beats quicker. And then where'd you go to high school?
Belvedere High School.
And then where'd you go for your undergrad and what did you study?
Northern Illinois University. I got my degree in biology with a minor in chemistry.
Very good. And I know that your band played at a person I know at his farmstead out in Shabana.
I remember it partially.
Yeah. And then where'd you go for your advanced degrees?
I went to the University of Arizona, where I studied at the Arizona Cancer Center.
And you got a PhD in?
My PhD is in nutritional sciences in the College of Agriculture, but my dissertation was in medicinal chemistry and natural products.
And then what were some of the places you were at between then and when you came to UW-Madison?
Well, after I left my undergraduate degree, I think I got a letter of recommendation from a Dr. Zinnan, if I recall correctly, and I went to work in the biochemistry department at Rush Presbyterian St. Luke's Medical Center in Chicago. I decided I wanted to go back to school. I moved out to Tucson, just packed up my stuff and threw it in a car in a 77 Buick LeSabre. Went out to Tucson and went door to door until I found a lab.
Very good.
And after I finished up my PhD there, I was there for 15 years. I also worked as a technician and a postdoc for a short time. I came to Madison. I worked at pharmaceutical products development in Middleton. Then I moved to Wasteman Biomanufacturing at the University of Wisconsin-Madison. And then I moved to help get the Program for Advanced Cell Therapies up and running.
Great. So tonight you're going to talk with us about living pharmaceuticals for first-in-human clinical trials. I look forward to your talk, and thank you very much, Ross, for sharing your insights tonight. You can take it away anytime you'd like.
Okay. Okay, is that visible?
It is, but we're also getting the next slide view. So if you want to, if you want to see if you can do,
because I'm looking at the okay, wasn't expecting that. And now?
Now we have a black. Now we're back to seeing your with the stuff on the left. Well, why don't we just go ahead with what you can do, and there you go. Swap duplicate.
See if that makes any difference.
There it is. That's you got it.
Okay, great. Okay, so yes, my talk is living pharmaceuticals for first-in-human clinical trials. As mentioned, I'm Ross Meyers. I'm the Director of Cell Manufacturing for the Program for Advanced Cell Therapies. We're in the School of Medicine and Public Health and within the division of the Carbone Cancer Center, or the department rather. I'm also a principal instructor in the School of Pharmacy in the Division of Pharmacy Professional Development and Applied Drug Development. So the University of Wisconsin Program for Advanced Cell Therapies is really a partnership between the UW Health Hospital and clinics and the clinical laboratories and the School of Medicine and Public Health. And what we do is facilitate UW researchers, clinicians, and external partners to try to translate innovative cell and gene therapies from the laboratory benchtop to the hospital bedside. So we are currently doing research in a research lab and translating those to our laboratory, where we can manufacture cells to be able to give those to patients. So our mission is to develop and deploy first-in-human UW medical science. We provide patient access to promising international cell therapy technologies and enable partners in industry and academic institutes to improve patient outcomes. Our long-term vision is to become the national leader in healthcare integrated personalized cell therapies to improve clinical outcomes. And we have pediatric studies as well as adult studies. So the program was started in 2016 when the School of Medicine and Public Health brought in Jacques Galipeau from Emory University in Atlanta. He's our program director, and he's one of the world's experts in cell therapy. Within our group, we're a lean group, but we are flexible. Within the group, Kimberly McDowell is an MD PhD in our program. She's the director of regulatory affairs. So she works with me to create documentation to communicate directly with the US Food and Drug Administration to prepare investigational new drug applications. Dr. Inga Hoffman is our medical director. Andrea Panatti is our associate director for research and development. He's developing the work in the preclinical research end of things. Crystal Weinberger is our program administrator. As mentioned, I'm the director of cell therapies manufacturing, and I oversee the manufacturing specialists, Olga Gantz, Nicholas Surprise, and Jenna Meyers. Nicholas will be leaving us to go off to law school next week and a master's degree in public health. So we are losing one of our people, but Jenna is stepping in. I also wear the hat of quality assurance in our group, and therefore I work with an independent team, which we call the quality oversight team. And they're in the UW Health clinical labs. They go through all of our documentation and actions throughout the year to ensure that we're following our standard operating procedures as per good manufacturing practices. That's Jinju Lee and Jill Palmer. So UW-PACT is a good manufacturing practices, or GMP facility. That means that we're regulated by guidances from the US Food and Drug Administration. And we're specifically, at least currently, only doing phase 1 clinical trials for investigational new drugs. Our clinical trials have to be approved by the Food and Drug Administration under investigational new drug application, which includes a clinical trial design, preclinical data, and chemistry, manufacturing, and control. The CMC is what I'm responsible for writing, and that's what we'll be focusing on today, is cell manufacturing. Being GMP means that all of our actions must be performed under standard operating procedures, and they're rather detailed standard operating procedures. All of our equipment and instrumentation has to be validated, and we have to demonstrate that the instruments and equipment are working to perform the functions that they are designed to perform, and specifically for our cell therapy manufacturing. In addition, our procedures are also validated, which means we perform our detailed procedures several times to ensure that we get reproducible results and the quality that the cell therapy needs to be used in the clinic. Our staff are all highly trained, and they have to demonstrate proficiency in all that they do. All of our actions are documented in real time and signed and dated. Any unexpected results that we collect are investigated and documented in real time and followed up on. This ensures the quality of our final products for patient safety. And we also do quality control testing to demonstrate identity, potency, purity, sterility, and safety. Our program isn't the only one. There are about 40 programs like ours across the country. The Food and Drug Administration categorizes cell therapy into two containers, if you will. There's minimally manipulated cell therapy and more than minimally manipulated cell therapy. So a minimally manipulated cell therapy means that the cells that you take from a donor are performing the same function in the recipient. But if you take the cells out and you manipulate them to do something specific or change them anyway, the Food and Drug Administration regulates that as if it were any other drug. Therefore, it must be under good manufacturing practices. Our facility is located within the UW Health Hospital, and this is for a good reason. If we were to be somewhere else in manufacturing cells, we would have to go through a lot of trouble through transporting and to ensure that the cells were at the right temperature. And we still have to do that in this little transfer that we do, but there's a lot more that would have to happen if we weren't integrated within the hospital. So now I'd like to present a video tour of the lab that my team created for you.
Hi everyone, my name is Nicholas Surprise, and I'm a cell manufacturing specialist with the University of Wisconsin Program for Advanced Cell Therapy. And here we are in the GMP cell manufacturing facility. Here at UW-PACT, the facility is comprised of 3 rooms. We have the gowning room here, through this door is the laboratory, and then after that a degowning room. I'm hoping to give you a brief tour of all 3 rooms, so get ready. Before we go into the clean room laboratory, we have to get gowned up. Normally when we have a patient, we have to wear sterile gowning. That means besides the regular gowning we would use, we have to go to another level of sterility. So normally if we had a patient that we were manufacturing, I would be in scrubs, but for right now, just use your imagination. Our sterile gowning procedure is a little complex. That's why we want to show it to you. It comes with the suit here, which you can see is vacuum sealed. There's a hood and some boot covers. So I'm going to put these on right now. I'll see you in a little bit. Okay, I've got my hood on and I've got my gowning on. The last step is to put on my sterile boots. But I wanted to tell you this. The reason why that gowning procedure is so specific is to make sure that none of our outside skin or any bacteria we might have on our body gets on the outside of this gowning. The outside is completely sterile. The inside touches our body. So we have to go through that procedure to make sure that we don't contaminate the outside. All right, now the boots. All right, I'm all gowned up. One of the big things we use here, one of the big supplies in our inventory, is IPA, or isopropyl alcohol. We use this to spray our gloves, spray any inventory we bring into the lab, to try and kill any bacteria that might be on it. So, before we go into the lab, I want to bring in a couple of supplies. We're a GMP facility. GMP stands for Good Manufacturing Practices. That means, generally, that a lot of our practices here are heavily regulated, and all of them are dictated by SOPs. That's standard operating procedures. That's a document that tells us exactly what we need to do, and we have to follow those documents every single time. We have SOPs for our manufacturing, for getting gowned, and even for bringing supplies into the lab. So I'm going to walk you through one of those SOPs now. Here is a supply I want to bring into the lab. I'm going to use that IPA, or isopropyl alcohol, to spray this supply to ensure that it's sterile. Now that it's set on the cart, I can bring it into the lab. Our lab is known as a clean room. That's an even higher level of sterility. So, some of the products we use are double or triple bagged. Everything within here is sterile. So, when I remove it from the packaging here, I know that I'm setting on the bench something that is sterile. It's as if I used a bunch of IPA, but instead this was irradiated. So I know it's sterile and I can bring it into the lab. Now, we're all set to head into the clean room. I'll meet you inside. Welcome to our clean room laboratory. This room is exactly that, a clean room. It's much cleaner than the room we just came from, and that's because of two things. First, we have HEPA filters that line the whole ceiling, 6 of them, that are constantly taking the air and exchanging it and recirculating. In fact, in 2 minutes, all of the air in this room is completely renewed by those filters, completely changed. This room is also under what's called positive pressure. That means that the air from here flows out to the next rooms, but the air from the next rooms can't flow back into this room. And that's because this room's cleaner than those two, so we don't want the air from there to come into here. So that's what helps make this room clean. When it was constructed, we also took care to use materials that would be resistant to the harsh chemicals we use when cleaning. So we have stainless steel surfaces, and our walls even use paint that resists those harsh chemicals. That ensures we can follow our procedures to make sure this room stays clean and lasts. Our GMP facility is heavily monitored. In fact, we use an automated monitoring system to check many different parameters throughout the facility, including temperature, the pressure differential that we talked about, and humidity. In fact, our refrigerator right here is monitored with the temperature probe that tells us exactly what the temperature is in that refrigerator at that time. And we have a predetermined range that we want to monitor. This system will tell us if the temperature in a piece of equipment like the refrigerator ever gets outside of that predetermined range. It'll send us an email, it'll give us a phone call, no matter the time, so that we know this is outside of its range. Remember, this is a GMP manufacturing facility. In this facility, we manufacture products, and it's GMP, Good Manufacturing Practices, which means it's regulated. One of the aspects of GMP that we haven't talked about yet is documentation. We document every action that takes place in this facility. For instance, here's a clean room notebook. These pages are specifically made for the clean room laboratory, so they don't shed as many particulates or particles as a normal piece of paper would if you, for instance, wiped your hand across it. This paper is made specifically for the clean room, and we write on them using specific clean room pens. So you can see, if I open this here, we document every action here. We also document things like equipment maintenance. If we have to calibrate a piece of equipment, we write down when it happened and what happened. We also document things like cleaning the biological safety cabinet, or BSC, that we use for a procedure, or even documenting the temperature or CO2 of an incubator. As you might imagine, one of our main goals here is to have control over all of our processes, and that extends to our equipment. This is a flow cytometer. We use this piece of equipment to analyze the cells we manufacture in this facility. When a piece of equipment gets installed in our lab, a new piece of equipment, it has to undergo processes called qualifications to make sure that it's functioning properly. And it doesn't stop there. We do weekly maintenance actions on pieces of equipment. We do preventative maintenance actions on equipment, which might happen twice a year or once a year. And to make sure that it's functioning properly throughout the entire day, we have the pieces of equipment, some of ours, hooked up to unlimited power supplies and connected to a backup generator in our facility to ensure that it's always ready to function. Manufacturing of cell therapies can occur using 3 different methods: manual, partially automatic, and fully automatic. Traditionally, manual manufacture of cell therapies happen in the BSC, and we still use this for the majority of our therapies. But if you were going to manually manufacture all your cell therapies, all of the actions would happen in this biological safety cabinet. The partially automatic way of manufacturing cell therapies uses an instrument like this, where you have a lot of actions that require manual effort, but a machine that uses valves and a magnet can do some of the actions. Finally, a fully automatic cell processing machine does all of the action inside in a closed system using the valves and different pieces. And we'll talk about this more next. Many pieces of equipment in our laboratory are very expensive. In fact, some pieces of equipment cost almost as much as an entire house. This piece of equipment, if you remember, is an automated cell processor. It functions as if it's an entire laboratory inside in a closed system. It has a centrifuge where cells spin around. It can control the temperature and raise it up and down. There's a microscope inside here. There's a pump that uses tubing to move all the cell product all around. A huge magnet. And we use this all for some of our cell manufacturing to make our cell therapies. Thank you so much for joining me on my tour of the GMP cell manufacturing facility. As you can see, a lot of resources and effort go in to ensure patient safety. And now, since our facility is guided by unidirectional flow, I'm going to exit via our degowning room. Bye!
All right, Ross, can you put your PowerPoint back up?
Yeah, can you see it now?
Nope, I'm seeing you full screen.
Oh, let me bring that back in here.
Okay.
How about now?
Nope, not yet.
Oh, I know what to do here. Just one moment, please.
Here we go.
All right, I think this is gonna come up. How's that?
We're seeing the next slide view again, but if you
Oh.
continue with that, that'll be fine. Or if you could
Oops.
remember what you did, just switch it around.
I can get it switched here. Just one moment. Sorry about that. How's that?
That's it. Way to go.
Okay. So now I'd like to switch gears and start talking about our active clinical trials. This slide is really here to give you an idea of what has to happen and for me to talk a little bit about the timeline that has to occur. Can you see my pointer?
Yes, I can. Yep.
Okay, thank you. So in particular, I'd like to draw your attention to these first 5 projects, which I'm highlighting right now with the pointer. And those are the ones I'm going to talk about today. But importantly, in this slide, from concept to developing an investigational new drug application and submitting it to the Food and Drug Administration can take 3 to 5 years. The approval process, if it's approved, doesn't take that long, but that isn't all that occurs. The University of Wisconsin also has an institutional review board that also has to approve and go through all the details of our study, and it's quite extensive. Once that's approved, we also have to go through a study activation, which can take a few months. And you can see by this, we've already treated some patients in these studies, and I'll talk about those more in a moment. And we have 3 additional studies that are just about to launch. So we're getting rather excited that we're going to be treating more patients here soon. So 3 of our cell therapies fall within something called virus-specific T lymphocytes. And I'll go through each one of them independently, but they're all somewhat related. So our first study we're going to take a look at is a phase 1 study for evaluating the safety and tolerability of virus-specific T cells against cytomegalovirus in adult solid organ transplant recipients. So cytomegalovirus, well, if your immune system is functioning well and you're a healthy person, you're probably not going to notice that you have that. It's going to be like a cold maybe. But when patients are taking immune suppressing medications, like people who have just had an organ transplant, these viruses cause great risk to sickness and even death in organ transplant patients. Similar study is for using the same cells therapy for patients who are immune compromised who have had bone marrow transplant. We also just got approval for a phase 1 study for safety and tolerability of these types of cells against BK virus in adult kidney transplant patients. And BK is another virus that you may have had and not even realized it, but if you're a kidney transplant patient, this virus can result in sickness and even death or rejection of a transplanted kidney. So as shown in the video, this particular cell therapy is somewhat automated, and we're using an automated cell processing instrument called the Clinimax Prodigy, which is manufactured by a German company called Miltenyi Biotech. And as you can see, and as Nicholas described, it's an entire laboratory kind of in this unit. This thing is an incubator and microscope. It has a heating and cooling element. Here you can see there's filters and reagent vials, a bunch of valves and a pump, the donor cells, media bags and wash solutions, a user interface, an electromagnet, it's rather powerful, and a barcode reader to identify all of the products that we have here, and a tube sealer for when we collect our cells. This is a schematic to try to give you an idea of what's going on inside of that instrument. So below you here on the left, you'll see where it says donor screening. And what we do is we test a close relative to see if they've had CMV in the past and recovered, or cytomegalovirus. We are also able not just to see if they have antibodies, but we can test their T cells to see if they can be reactivated. Also, the donor can't be sick. They have to be free of active virus, because we remember the patients are immune compromised. So we can't give them even yet another virus. So we do some preparation in the lab, but the instrument starts to expose these memory T cells. And what do they remember? Well, they're remembering that they were exposed to cytomegalovirus in the past. And so there are some proteins, or rather peptides, that are added that are common to cytomegalovirus, and they start to reactivate these cells. At that point, the CMV antigen is washed away, and then the T cells are reacted with an antibody to a cytokine called interferon gamma. And that's what this diagrammatic is supposed to be showing, that those antibodies are on the cell surface bound to interferon gamma. When the cells become activated, they start to secrete that cytokine interferon gamma. That's what these little green diamonds are for, if you can see that. Then the instrument's going to add yet another antibody that binds to interferon gamma, but on the antibody, there's a tiny ferromagnetic bead, or a magnetic particle that's reacted with, and those will only bind to the cells of interest. So an electromagnet is turned on, and all the cells pass through a steel column that's magnetized. The cells of interest are going to bind magnetically to that column, while the unwanted cells pass right through there. Then the magnet's turned off, and the cytomegalovirus specific T cells are collected. This process is called magnetic activated cell sorting, or MAX for short. So we're not quite done yet. We still have to do quality control testing on the final product and make sure that the cells pass their quality specifications and ensure that they're safe to give to the patient. If they are, we put them in a final formulation. This whole process takes 2 days. So moving on to another active clinical trial, we have T-cell depleted bone marrow transplant. TCR stands for T cell receptor alpha beta. That's a type of receptor on the T cells. CD19 refers to B cells in your immune system. So we are going to remove those cells to, well, I should say this therapy is a specialized type of bone marrow transplant that's designed to prevent graft versus host disease in children and young adults who have non-malignant bone marrow failures or immune deficiencies. So it's a very special type of bone marrow transplant. We use a semi-automated process that removes the T cells that are responsible for graft versus host disease prior to transplant into the recipient. A lymphocyte donor is identified, and they give a very special type of cell collection. It's called mobilized leukapheresis, and this can only be done once in a lifetime per donor. So there's a little bit of pressure here to make this work. In addition, the team has to be in the clean room for 13 to 14 hours. We are able to, if we have to, leave for food and water and a bathroom break, but for the most part, we're in there performing continuous actions that entire time. There's that semi-automated instrument, and also this is manufactured by Multani Biotech, and it's called a Clinimax. So what happens here is you have the donor blood cells and some washed solutions and media, but the cells are initially prepared for processing, and we put them on the instrument. The T cell receptor alpha beta and CD19 cells are bound to an antibody, to antibodies with those ferromagnetic particles. The cells magnetically collect on the steel column in that magnetic field, or the magnetic activated cell sorter. The cells that are bound, the cells that are not bound pass right through that magnetic field. these are actually the cells we want. We're collecting the cells we don't want on that steel column. Once we have the cells, we again have to perform quality control testing to ensure that the cells are what we think they are and that they're sterile and safe to give to a patient. If they pass the specifications, they're then prepared for patient administration in a final formulation. Something in the way here from Zoom, and I can't quite get rid of that. But anyway, the next clinical trial that we have that's going to initiate very likely next month is mesenchymal stromal cells for radiation-induced xerostomia. So the name of the study is a phase 1 study for autologous. That means the patient is going to get their own cells. Autologous mesenchymal stromal cells in patients with xerostomia after radiation therapy for head and neck cancer. So xerostomia is a consequence of long-term consequence of head and neck radiation therapy, and it really does significantly impair the quality of life in these patients. They get radiation-induced xerostomia, and it can develop dental caries. They have a impaired swallowing ability, so it's difficult to eat, difficult to speak. They don't taste food the way you might do if you don't have that, and oftentimes it results in poor nutritional status. So this cell therapy is rather exciting for us because Dr. Gallipo is one of the world's experts in mesenchymal stromal cells, and this is a regenerative medicine approach. The MSCs have the ability to promote tissue healing. That means that basically biochemical signals sent from the MSCs stimulate the self-renewal ability of the salivary glands. This is a homegrown technology here at the University of Wisconsin. This study will be the first in human and the first in the world. So this is rather exciting for us. In addition, the principal investigator on this study is Randy Kimple, and he just was awarded a rather substantial grant to continue this study in people. So the manufacturing process by which we do this, I'll break down one step at a time. So bone marrow aspirate is collected from the xerostomia patient and transferred directly to the packed clean room. We do a lot of paperwork, but the next thing we do is we use gradient density centrifugation to isolate mononuclear cells, or the cells of the immune system, basically. Those cells are incubated in culture flasks and media for 72 hours. And the MSCs will adhere to the plastic culture flasks while the unwanted cells will not. So this is a form of separation and a form of purification. We remove the unwanted cells by simply aspirating them out of the culture flasks. The mesenchymal stromal cells are allowed to proliferate until there's enough of them for a therapeutic dose. They won't proliferate endlessly. They will eventually start to slow down in their growth and division, but we think that we will be able to get enough of those to create a cell bank to continue. And we've done this several times now, and we know that we can reproduce it. The MSCs are then stimulated with the cytokine interferon gamma for 24 hours. They're then harvested and we count them. Some of the cells are used for quality control testing to ensure that they pass the quality control specifications for identity, purity, etc. The rest of the cells are cryopreserved in liquid nitrogen, and that's quite a process just in and of itself. When the cells have passed the quality control specifications and the patient is ready, the cells are thawed and incubated overnight. That's a process called culture rescue. Kind of gets the cells, wakes them up from cryopreservation. The cells are then prepared at the correct dose and placed in final formulation for administration in the clinic. When we hand it off to the clinic, a surgeon will inject the interferon gamma stimulated mesenchymal stromal cells into the salivary glands by syringe. This is a diagram showing you where those glands are. So this is a summary slide. PACT is, to summarize, PACT is a partnership of the UW School of Medicine and Public Health and the UW Health Hospitals and Clinics and the clinical labs. PACT is a good manufacturing practice facility and operates under the United States Food and Drug Administration regulatory guidance. We have 5 active drugs, investigational new drug studies in progress or about to initiate in the next few months. Cytomegalovirus stimulated T lymphocytes for solid organ transplant. Cytomegalovirus stimulated T lymphocytes for bone marrow transplant. BK virus stimulated T lymphocytes for kidney transplant patients. T-cell receptor alpha beta and CD19 cell depletion for bone marrow transplant. That's the one that's designed to prevent graft versus host disease, especially in children. And interferon gamma stimulated mesenchymal stromal cells for radiation-induced xerostomia in head and neck cancer, or xerostomia or dry mouth. I'd like to acknowledge both Nicholas Surprise and Olga Gans for their excellent video giving a tour of the clean room. And I'd like to thank Dr. Jacques Gallipeau for slides he lent me and his continued support in putting this together.
Thank you, Ross. Are you ready to field some questions?
Absolutely.
Well, people can either chime in anytime you'd like and ask a question.
See, there's something in the chat box. I'm not sure what that is, but
yeah, Kathleen Ott and I were both wondering about this. Why is mobilized leukapheresis something that you can only do once in a lifetime?
That's a really great question. I had a
bizarre.
feeling somebody would ask me that. So they give the donor GMCSF, and I'm going to have to now, granulocyte macrophage cell stimulation factor. That will mobilize cells from bone marrow. And what it does is enrich the donor's blood with immune cells and stem cells for the immune system. So it's not something you want to have done multiple times throughout your life, because it is rather not invasive, but it's that bringing all of those cells out prematurely is probably something you really only want to do once.
Okay. Larry Winkler is asking, what is the multiplication factor of MSC proliferation?
So we actually measure that by doubling time. We don't do it by what some people would consider passage number, the number of times the cells pass, but you can't go, I think, for MSCs beyond 50. Oh, I'm not sure how many doublings it is now. I know that's an important fact, but I'm not sure how many doubling times we can go out to. But we know thus far we haven't come anywhere near the limit.
Other questions that people might have?
This is Larry Winkler. Do you have a sense? You're mentioning something like 50 doubling times?
I don't think that's right. I think it's less than that, but I don't know the exact number off the top of my head, Larry. Sorry.
And then Ross, if you could stop your screen share, then
Sure.
better view for Q&A. Thank you.
Let's
Thanks,
see
Tom.
here. I think I just maybe need to close out the presentation like that.
Let's hope so. Or if you stop if you stop screen share.
Stop share, maybe.
There you go. You got it. Great.
Ah, there we are.
So what are the mechanics of this? You're in the hospital building. You took Liz, Jesse, and me through a tour. You can have patients several hallways away, but you don't have to go outside. It's right there. You're wheeling carts, not driving Ubers or.
That's right. And in fact, as mentioned, everything we do is controlled and regulated. So when we finish a product, we put it inside a validated cooler with gel packs. And inside there, we also have temperature monitors, 2 of them, in case one fails. And then we also have to hand off the cooler to the clinical team, and there's several signatures. And oftentimes the clinical coordinator will accompany us from the clean room, the exit of the clean room, to the clinical trials clinical trials area of the hospital, where they inspect the appearance of the product. A medical doctor, the medical director, and a nurse all need to sign off on that, as well as I do. I have to certify that all the quality control specifications passed, and to the best of our knowledge, it's safe.
And so I'm assuming at this time the patient is all set and ready to go, prepped in whatever kind of surgical suite you're in. So this is kind of like an organ donation. It's got to be orchestrated.
Well, it's interesting that you mentioned that. We are about to start a project with a Madison company called Opsis, where they'll be replacing human retinas. And that will happen in a surgical suite as well. The lymphocytes that we manufacture, the viral specific lymphocytes, those are intravenous injections, and it's done right in a regular hospital room. But of course, the injection into the salivary glands is done by a surgeon in a surgical suite.
Okay. Even though it's just an injection, it's considered a surgery?
I think there has to be a sterile environment, at least somewhat, and the patient likely needs to be addressed, although I don't know that part of the process right now.
I wanted to chime in on the xerostomia because my dad got throat and neck cancer in in 2009. By March of 2010, he had died of it. And one of the things that I really remember was that whole thing, the thirst, the swallowing. There were a lot of unpleasant things about getting cancer and going through a 6-month demise. But boy, when I saw that Randy got that grant, Randy and I had children together. Well, not together. Our 3 sets of children. Sorry about that, Randy. We're at Bernie's daycare together, and so it's been interesting to watch his research and that of his wife. They both have their separate research labs, but that one's really close to home for me, and I hope.
Yes, that grant will cover the cost of the entire study. So we're very excited about it. All the pieces fell into place very well on this one.
And we didn't even have, I'd never heard the name Xerostomia until I saw the news that he had won that grant. So
yeah,
that one I'll be watching because I do have a dog in that fight. So that'd be great. Let's see if there are any other questions. How can you replace a retina and get it wired up?
So that I don't know. But I will tell you that technology was developed at the University of Wisconsin-Madison at the Weissman Center. That is in a large collaboration with Fujifilm Cellular Dynamics and a couple of other companies. And I don't know how that surgical procedure is performed. Our responsibility in that, and I mentioned that part of our mission is to facilitate any work we can do to help these therapies get into clinical trials. So it turns out that a company can't just come in and operate within the UW Health. They have a lot of rules, and we have a lot of GMP rules and a lot of controls. So I'll be meeting with them tomorrow. We're going to discuss how the protocol, once the cells enter the hospital, what's going to happen after that. I think there's probably going to be some sterility testing potentially. We're not sure. We're going to talk this out tomorrow, but we have to develop a protocol, a detailed protocol for every step that happens along the way up until the retinas get into the surgical suite. So that's our role. The name of the company is Opsis, by the way, Opsis Therapeutics. You can go to their website.
And Diana, I'll see if we can get Wednesday night at the lab on that.
David Gam is the
Yep, that would be
leader on that. Yep.
he's
that
done
would
at
be a
least
great
2
one.
Wednesday night at the labs for us over the past 15 years. And if he's ready to go with transplants, that would be great, because that's
Very, very exciting, especially for here in Wisconsin.
good. Dareth is asking, when the desired cells are the ones with iron attached for magnetic sorting, does the iron need to be removed before the patient treatment with those cells?
I'm amused that was the first question I asked when I saw this technology. And the answer to that is no. But I don't, nothing, I can tell you that there's no further processing after the cells are released from that magnetic steel column, that magnetic field. There is nothing else that happens after that. So my assumption is that those antibodies bound to the ferromagnetic beads are still there. But I will say that this technology has been developed over the last 20 years, starting in Germany. And there hasn't shown to be any patient adverse events as the result of that. I think one thing to maybe realize is how very tiny that bead actually is. I think it's hard to maybe, at least for me, to get my head around it.
Well, I'm reminded that your little pellets of iron and some iron supplemented breakfast cereals. That's one of the things, you have the kids crush it up and then you use a magnet and you pull those things right out. So that doesn't prove that it's not an issue, but it's like, wow, what would make it dangerous?
Yeah.
You'd have to test that, but Dareth and I are both old enough to remember the ads for Geritol, so no more iron, poor blood.
This is a bit of a nitpicky terminology question.
Sure.
I noticed in your video, you talk about spraying things with isopropyl alcohol and describe it as sterilization. I've worked in a few projects involving sterilization of and high-level disinfection of medical instruments. And in that context, something like that would probably be considered disinfection as opposed to sterilization.
Right, it's an aseptic process, right? The part that he didn't mention is that we also use solution called that contains peroxide. So the whole laboratory is not only cleaned with isopropyl alcohol, but the whole laboratory is cleaned with a peroxide solution.
Okay.
So that kills just about everything you can think of. But yeah, good point. My team put that video together last week in about 2 hours. I was really rather impressed with how they did that. Nicholas has some experience on the stage, and it really paid off this time.
Very, very, very well produced.
And who did the editing?
He also did the video editing.
And he's going to become a lawyer?
He has I think a pretty full ride scholarship through law school and the Master in Public Health program. So he's a clever guy in many ways.
That's excellent. Any other?
We're sorry that we're losing him.
Yeah. Additional questions for us? What can you tell me about the funding for the overall pact? If we had a pizza pie graph, how big is your outfit and where do your various sources
So
come from?
right now, we're operating on the initial costs that were brought in when Jacques Gallipeau was brought in to set up the program. Now, keep in mind that the UW Health has committed a lot of funding to the construction of that clean room facility. It's right next door to the bone marrow transplant facility program. So just the, I don't know what the price tag was on the clean room, but I can tell you just to keep that thing maintained, the filters, the monitoring system, all of that is very expensive, and the commitment from UW Health is really rather enormous. Having said that, we were able to negotiate them building us a quality control laboratory as well, and a storage facility and some quality assurance computers in space, which was approved for this fiscal year at around $247,000. The laboratory itself, I'm sure, was over $1 million. The startup funds were in the neighborhood of $5 million. We haven't gone through that yet, but that's very likely because COVID slowed down our progress in terms of being able to treat patients. So the performing clinical trials and the clinical trial suite was not a priority. And there's a lot of work that goes on to get eligibility for patients and for donors. And that requires a lot of people and a lot of time. And so those people were working from home. And I think all in all, there was I guess I could mention that there was a large COVID-19 vaccine study being performed in the clinical trials group. And so that consumed all of their time over most of, half of 2020 and well into 2021. It's still ongoing.
And then Larry just asking, what's the expected sample size for phase 1 trial of this nature?
That's a really good question. So for the virus-specific T cells, we would like to be able to treat 20 patients in each one of those studies. There's 3 studies that takes you up to 60 patients. I believe the mesenchymal stromal cell study, we intend to do 12 patients initially. There is some dose escalation. So when if we can show safety at a lower dose, we'll manufacture a greater number of cells and increase the dose to demonstrate that the therapy is safe. That's true also in the cytomegalovirus study, but not in the BK virus study.
And then what's your capacity along those lines? If you're looking at 12, 20, 60, and you have a process, at least in one case, was a 2-day cycle for one patient.
Well, this is a really great question, Tom, because the part that, I did mention that there's a lot of work that has to happen in the clinical trials group to show donor and patient eligibility. And some of the patients in the cytomegalovirus study are taking a drug called ganciclovir, which actually does work against CMV, but their levels, some can't fight it. And so the levels kind of go up and down. So eligibility changes. But because of all the paperwork that we must prepare, we have to order special products that expire really quickly. And so the logistics of doing this is quite complex. And so I have to issue all of the paperwork and all of the forms. And when we're done, the batch record is the size of a book for all the recording that we do. So there's a lot of recording that happens during the process, but there's a lot of recording that happens afterwards, testing results. I have to go through all of that. So this is a great question because we're talking about it right now. How many patients can we do in a given time? We right now realize that because we just treated a patient 2 weeks ago, we're realizing that we probably could treat one viral specific T-cell patient in a 2-week period. Keep in mind that we have to purchase those materials from Germany. So they can get it to us overnight a lot of times, but sometimes they can't. So like I was saying, the logistics of all of this, Olga Gans has gotten very, very good at working with these companies, getting these reagents that expire quickly, so we don't buy them and then the study doesn't happen. We're talking thousands of dollars for these reagents. So there's a little bit of a trick there. The mesenchymal stromal cells are less expensive to manufacture, but we're committed in more time. So we can probably only really do one patient a month, I'm thinking at this point. So, and again, we don't have a lot of people working in there. We don't have a giant clean room or multiple clean rooms. So we are going, and we have to clean the entire clean room and do changeover every time we do something. So we do anything in there, we have to do a changeover. If we can't do 2 cell therapies at the same time, we don't have that capacity in people, but we also, you have to put a lot more controls in place to make sure that your cell therapy is going from the right donor to the right patient. There's a lot of labeling and a lot of actions that have to happen. We do have quarantine of all of our materials and release, and we keep tight control on expiration dating. So the answer to your question, Tom, is really complex, but that's kind of what we're thinking right now in terms of our capacity. We are talking about switching the program over to an institute, which would potentially be able to help us hire more people, bring in some funding from other sources, charitable sources, for example, philanthropy. And so that's what's going on right now. We know that we're about to get a wave of patients, and the logistics of that are going to be challenging, but we're up for it. This is going to be great.
Yeah, the bottleneck management is really impressive, because especially when you can only have one bottle at a time and not a 6-pack or a case.
Exactly.
And I'm not enough of an engineer to know how you do that, but I can anticipate that that's a lot of extra work figuring out how to go from n=1 to n=3 to n=9.
Yes, so scale up for us is very limited.
Thanks, Larry, for the question on sample size. Are there any other questions that people would like to ask? You can chime in like Wynn did and I did, or you can put them on the chat. And if people want to watch more about your program, PACT has a website, has a Facebook page, so folks can go there.
We have a website. I don't know if we have a Facebook page. I know
Okay.
Jacques Galipeau posts a lot on LinkedIn.
Okay.
But we also, from time to time, we do have some of internal news reports come out. I think you may mention that you saw something that with Randy Kimple. So that was a great way to be able to follow us. But our website, we keep it updated regularly, and it'd be great if people could continue to follow our progress. We think we're starting to develop with the Institutes for Discovery. We're just getting a gene therapy program up and running with Chris Saha's group, and that's going to be it's a CRISPR project in gene editing, so that's going to bring us to another level as well.
So Dareth, if you want to read that, well, I'll read that. Excuse me. Dareth says, so you're totally at the bleeding edge experimentally, but hope eventually to spin this off to a larger organization to scale up potential treatment numbers. How long will that take? Are patients queuing up for these treatments?
The Xerostomia study is as far as from what we know, there are several patients that are pretty much waiting for this clinical trial to launch, and we're getting very close. We think probably most likely in August, which is, right around the corner. But the question of being able to scale up, so that is one of our goals. We want to be able to make this available and what Dr. Gallipo would call a service line. Now, we're not going to be able to necessarily do a whole lot of that type of work. We think we may be able to in the future, but the idea here is that we would find an outside partner that would be interested in creating a GMP clean room somewhere, maybe in Madison, and be able to treat patients. So we would do what we could to facilitate and translate the technology to a program that could do these cell therapies on a larger scale for a greater patient population. So that is something that that's in our intentions.
Great. Thanks, Dareth, for the question.
Yeah, great question.
Any other questions before we head off? I'm looking here at the sky. The sun is already down behind the building. We're 5 weeks after the summer solstice, which is the sun was still up above the building 5 weeks ago, and it's already down. And I'm going to go home and batten down the hatches.
Yeah, time to pull my tomatoes in.
Yeah. So look forward to seeing everybody next week for Corey Zetz's talk on the Menominee Valley Partnership and transforming the Menominee Valley River both as a sustainable environmental restoration and an economic development project. And then in 2 weeks, we'll have PJ Leash with the Insect Diagnostic Clinic here Wednesday night at the lab. Thank you very much, Ross.
You're welcome.
Really appreciate it. Good luck with all the projects that you're doing. And then the most important thing is tell us where your band plays next and when.
I'll be playing this next Sunday at 10 a.m. for the farmers market in Paoli at the Old Mill Park.
Very good. 10 a.m. on Sunday at 10 a.m. in the morning, as we like to say where I come from.
You can find us at the Radiant Beings on our Facebook page.
Very good. All right. Thanks, everybody. I'm going to turn the